right, good morning everyone. So today we're starting on chapter 3. And as I mentioned at the end of last time, we're actually moving through the topics a little bit faster than intended, which is great. It's probably going to mean that we end up with a little bit of extra time before that midterm. So we might have a discussion about what we've covered so far and things like that, but I'll keep you posted on our progress. So for today, we're starting with discussion on the organization and functions of the nervous system. So this is basically meant to be a roadmap for the rest of the semester, where we start looking at where things are and the basics of what they do, so that as we move on into later chapters and start talking about specific things that are happening, and where those things are happening, we have that baseline understanding. So this is fairly familiar, especially if you've taken Psych 104 recently, where you cover the basics of the nervous system. That's going to be mostly the same information. And then, as I said, we'll add complexity as we go through the semester. And so we're going to start with the central nervous system. So we already understand how the neurons themselves function and interact with each other, but now we want to look at their groupings. How are they organized and what do they do on that larger scale? So we are going to start by looking at the first of our two divisions of the nervous system. So you probably remember the central nervous system and the peripheral nervous system. And you'll sometimes see them noted as CNS and PNS. I usually just say the full names because that works with my brain a little bit better. And then once we wrap those two broad topics up, we'll spend the end of the chapter talking a little bit about things like development in the brain and how those structures come to be where they are. But as I said, starting off with our central nervous system, and this is such an extensive topic that we won't even finish all of the central nervous system today. We're just going to start establishing some terminology that's going to be very important, and then hopefully we'll wrap up a little bit early. So our central nervous system is basically everything that includes the brain and spinal cord. So everything within our brain, everything in our spinal cord is our central nervous system. And simplistically, everything that is not the brain and spinal cord is our peripheral nervous system. Before we get into it, I want to sort of point out a couple of terms that tend to get confused. And this is one of those things where psychology, and I suppose this comes from biology, have very arbitrary naming systems, we can say. So you will call the exact same thing by a different name depending on its location. So this starting point is meant to clarify some of that stuff and reduce confusion later on. So the first of these is similar words that do not mean the same thing. The difference between a neuron and a nerve. So we've been talking about neurons. Those are the single neural cells. Those are the individual units that we've been looking at so far. But we can then talk about nerves, which are bundles of axons that run together. So nerves have the axons from multiple neurons all wrapped up together. Now this is where that technicality comes in. The term nerve is actually only used in the peripheral nervous system. So we would never talk about a nerve in the brain. That's only for the external stuff or the stuff outside of our central nervous system. Instead, we can call the equivalent of nerves, so those bundles of axons running together, when they occur in the central nervous system, then we call them a tract. And so tract and nerve mean the same thing, but the name specifically tells you where it's located. So there is a conveniently handy table from the textbook that helps with this. So if we're talking about bundles of axons, so those are the sort of tail bits that stick out off of our neurons. If we're talking about peripheral nervous system, those are nerves. Central nervous system, those are tracts. Same concept, different names based on location. Okay, so now that we've established that this is something that biologists have decided is a good idea, we're going to look at another thing where they've done the exact same process. And so this is looking at groups of cell bodies. So that soma of our neurons, that is grouped together and named something differently, whether you're in the central or peripheral nervous system. So in the central nervous system, that group of cell bodies would be called a nucleus, which of course is also the name of a part of a cell, but hopefully by context we'll keep those separate. We can also talk about groups of cell bodies in the peripheral nervous system as ganglion. So again, with our handy-dandy little chart, we have the fact that we have two descriptors that have very different names depending on the location. So I'm going to try very hard to watch what I'm saying, and this is sort of our guide to go from here. So just our clarification out of the way. All right, so now that we've done that, we can move on to actual structures. And we're going to start by talking about, I think we're starting with the forebrain here. We're going to talk first about the cerebral hemispheres, and then we'll sort of move on into other major structures within that part of the brain. We're going to look at a lot of images and sort of talk through some of these things very slowly. There's quite a bit of terminology though, so again, when we have these sort of info-heavy lectures, I try and keep it to minimal topics at once. So when we talk about our cerebral hemispheres, and I'm going to hop between our text slide and our visual slide, these are the sort of large, wrinkled structures that dominate the brain's appearance. So if we go to our next slide, that would be the two parts of our brain. What we see on the outside are our cerebral hemispheres. And we can call this the dorsal or the superior part of the brain, and they're covered by the cortex. So that's that outer layer sort of at the top front-ish of your brain. We might also talk about the longitudinal fissure, which is that line that runs from sort of the front of your brain to the back of your brain, and it separates both of those hemispheres. So that's that line that runs down the middle there. So in this image, we're looking at two cerebral hemispheres separated by a longitudinal fissure. All right, and the two hemispheres are nearly mirror images of one another. They tend to have fairly similar divisions of function, but we do also see that there are some asymmetries. So each hemisphere tends to receive sensory input from the opposite side of the body, though it does still receive a little bit of input from the same side of the body. This is when we start seeing the first hints of that hemispheric lateralization, where one side does one thing and the other side does something else. So here we have that little bit of asymmetry where the right side of our brain tends to get information from the left side, and the left side of our brain tends to get information from the right side. All right, so we also can talk about some different structures or descriptors of parts of the brain here. So we can talk about a gyrus. So each ridge in the surface of the brain, so each of those curves would be a gyrus, and I believe the plural is gyri. So instead of the US, an I at the end, which is what is in the next slides. Perfect, or the next terms. We can talk about a sulculus, or again, I think the plural is sulculi, where we have the grooves or the spaces between those two gyri, the two bumps. So I think I have an image here. Perfect, yeah. So we have those kind of like hills. The bumps on the outside, those would be the gyri, and then the valleys in between, those would be the sulculi. And we can talk then about a fissure. So instead of just the sort of smoothed curve that goes inside, a fissure would be a more pronounced or a larger groove or space that occurs between two gyri. So it's kind of like a more intense version of the sulculus. And then we can talk already a little bit about the cortex. That's just the outer surface of the brain. And the root word for cortex literally just means bark. So it's meant to be that outer coating on the brain. And it's mostly made up of the cell bodies of our neurons. And those cell bodies tend not to be myelinated. So if we talked about the myelin sheath, that only really exists on our axons. So if the outer layer is mostly those cell bodies and it doesn't have lots of that white, fatty myelin sheath, then it's going to look gray. And that's where that part of our brain gets the term gray matter. And so this cortex is really, really associated with cognition because all of our neural processing occurs where our neurons are synapsing onto each other. And so that's where our dendrites and our cell bodies are receiving that incoming transmission from the ends of other neurons. Get it right? And so because that's where all of our cell bodies are located, that's where a lot of that contact is happening. So that's why they associate that with lots of cognition, because that's where a lot of processing is going on. And just some basic info about our cortex. It varies between 1.4 and 4 millimeters thick. I don't really care about the specific numbers here, but I just want you to note that that's fairly thin. And instead of evolving to have a thicker cortex, which we might expect if we wanted to have more processing power, what we've done instead is seen this evolution towards having convolutions. So having those gyri and sulculi, those ins and outs, that allows us to increase the surface area, which then gives us access to more of those cell bodies. If we had gone the other route, where instead of getting lots of curves, we went for a thicker cortex, we would have had to have a lot thicker of a cortex to get the same kind of area. And having a thicker cortex actually provides problems because it's harder to get to the cell bodies that are in the middle of that layer. So if we went thicker, it's actually harder for communication to happen. So these convolutions, these bumps on our brain, have actually evolved to allow us to have the maximum amount of contact between neurons in the most efficient manner. Yep.
I'm just curious, so if we're looking at like a fissure or a sulcus, is it possible for a neuron like on one side of like one wall to communicate with one that's on the other side of the fissure?
I don't believe so. We'll talk a little bit about it, but what seems to happen is, so the outside is our cell bodies, and then the inside where our white matter is, that's where the axons are. And so they tend to do the communication on the inside, and then they sort of touch out onto the gray matter. So we think of the brain sort of outside in, but the brain actually functions inside out. So the communication is sort of traveling information inside in that white matter, and then it touches out into the gray. And so we actually have like bands of axons that kind of reach out to different places. So there's typically not a whole lot of communication across gaps, but I don't think it's unheard of necessarily. I just don't think it's as common as typical communication. And I guess kind of from that, with the axons, I mentioned before that axons tend to be myelinated, and myelin is that sort of white fatty substance that wraps around the axons and lets them communicate faster and insulates them from the surrounding environment. So that's why that interior of our brain is called white matter, because it's all, or mostly, myelinated axons, so that part of the neurons that's surrounded in fatty tissue. And that allows that communication to happen sort of on the core, on the inside of those surfaces, which is really neat. And so if we look at our image here again, we see those outer parts, the cerebral cortex, that's our gray matter. The inner parts, that's the white matter, that's where the axons are sort of moving information around. And then we have lateral fissure, longitudinal fissure. So lateral fissure is sort of a deep crack on the sides of our brains. The longitudinal fissure is the one that runs along the sort of top of our brain. And then we have sulculi in between. So we typically only have the big fissures, the ones that are labeled and named, and we'll use those as reference points. and the rest of them are just the sulculi. Okay, and I think that's what I wanted to say there. The next thing we want to note is that it's not just sort of a free-for-all within the brain. There are sort of hierarchical organizations. So we can look at layers of organization as well as columns of organization. And that's what this image is meant to show us here. So A shows sort of those horizontal bands. Those are different layers within the cortex. So the cortex of most mammals, including humans, typically has 6 layers. Though in different parts of the brain we might be missing some, the overall is 6. And so these layers tend to be separated by fibers that sort of help supply nutrients and resources to those cell bodies of the neurons that are in that part. So they're kind of divided off so that they can be serviced in between, kind of like feeding off of roads in a city. So the roads are the divisions and they provide whatever shipping you need. And so you have different types and sizes of cells, which is why they tend to look a little bit different, and they tend to specialize for different roles. So we can look very, very briefly at what those roles tend to be. And this is another one of those where we'll talk very, very briefly now about it, and then go into much more detail when it's relevant later on. But layer 1 tends to be inhibitory, so signals there try and inhibit or prevent response. We have layers 2 and 3 are involved in associations. So if you're trying to remember the name of a song, you're trying to connect the sound and the title and maybe the artist, all those informational pieces are coming together. That happens in those associational layers. Layer 4 is sensory, so that's where we're getting that input of signal. And layers 5 and 6 seem to be involved in motor function. And again, not all of the brain is going to have all of these layers everywhere, but these are sort of the general rules. And then if we wanted to talk about our columns, so if our layers are those horizontal stripes, columns are going to be the vertical stripes. And so the cells that are in that, that did not go the way I wanted it to. All right, so the cells that are grouped or that are in that cortex get grouped into columns, and they run sort of perpendicular to the cortex, and they sort of serve that vertical function. It allows for a vertical transmission or communication between those layers. And then again, we'll come back in more detail to these topics when they are relevant, but this just gives you an idea that it's not a free-for-all in the cortex. There are structures. And interestingly, this is sort of where we start meeting that edge of what we know. So there's a lot of unknowns when we get into sort of what is each area doing or how exactly is information passed. This is where it's sort of theoretical and we're not positive, but we're starting to get a clearer picture. All right. So the next thing I want to do is take a brief pause and talk about brain size and intelligence. Now, mostly people have this underlying assumption that having a larger brain should correlate with being smarter. So larger brained creatures should theoretically be smarter. But there are a lot of species that have much larger brains than us. What seems to matter more is actually the comparative body size to brain size ratio. So if you have a large brain for your body size, then that would be a sign of intelligence. But if you look at very, very large species, they might have comparatively smaller brains for their mass, but their brain might still be much bigger than ours. So if we look at something like blue whales, they are massive creatures and their brains are fairly large, but the brain to body ratio is comparative with humans. So it's not like they're super intelligent creatures because they have huge brains. It's they have huge brains because they are big creatures. So what we really care about is that brain to body ratio, and species that typically have a larger brain for their body size tend to be quite a bit smarter. Now, this is sort of a caveat to our caveat here, but intelligence is one of those really confrontational topics. There's a lot of debates about how you evaluate intelligence. You might have seen the cartoon where they have a fish and a bird and a monkey, and they're standing at the foot of a tree, and they say that we're going to test your abilities, so climb this tree. And not everybody is going to perform the same way. That's kind of how evaluating intelligence works. Most intelligence-based tasks are biased towards what humans consider intelligent behavior. And so if you look at things like cephalopod research, as we start looking at octopi or octopuses, they tend to be very intelligent but don't necessarily show intelligence the same way that we'd expect in, say, primates. And so we have this issue where species that we had dismissed as being non-intelligent are showing a lot more promise and cognitive ability as we adjust our standards for gauging intelligence. So that's a whole topic. But I've done a seminar class where we've talked about it, and it's a really fun topic. So yeah, intelligence is one of those things where we have to be careful how we measure it. And I think in the next couple of years, we're going to be surprised what species start showing up as having intelligence, as the media will probably call it. All right, so that's the whole brain-body ratio. And then even within brain size, if you have a consistent brain-body ratio, there's another indicator that will usually correlate pretty well with intelligence, and that's complexity. So when we look at the human brain, we have a fairly large sort of forebrain. That's what it is. Having an issue with words today. Our forebrain is comparatively larger than our midbrain and our hindbrain. And the forebrain is what's associated with a lot of sort of cognition, complex tasks, comparing things, decision-making, all of that. So having a large forebrain, and especially for us, having a forebrain that has lots and lots of surface area, because of all of those convolutions and curves, that means that we have a very high processing power. So if we looked at similar sized brains that are less convoluted, if they are flatter, they might not have the same processing power. And there are actually, I think the textbook chapter starts off with a story of a woman who was having seizures and went for a brain scan to see what was going on, and they found out that her brain was completely smooth, which it was surprising, not that her brain was completely smooth, because that is a mutation that can happen, but the weird part was that other than the seizures, her life was pretty much normal. And not having all of those convolutions in the brain should reduce cognitive capacity and make it harder for her to function normally in society, but that wasn't an issue for her. So that's one of those case studies where she kind of went against what we assume to be associated with intelligence and processing power. But even between species, if we start looking at how convoluted or how much surface area different species' brains have, those with more surface area tend to be on the smarter end of the scale. Yep. I'm not really sure. I'm assuming that she's one of those people that they're studying very closely, and when she dies, they will study her brain even more closely, as is usually the case with case studies. But it seems like she just sort of overcame things by working harder and sort of making connections different ways. But even the textbook wasn't quite sure how this happened. And that's why it's such a weird situation, because from what we understand, that shouldn't have been possible. So that's, again, that edges of what we know and understand, and I'm sure that there are lots of people asking exactly that question. Yeah, it's unfortunate, like, for sometimes things like that, I'm like, I wish I knew the answers. But someday I hope. All right. Right. The next thing I wanted to do was talk about a couple of directional terms. This is more for utility than for something that you are really, really going to need to know in isolation. These are a lot of anatomy type terms. If you've taken an anatomy class, you've done these before. But I do like to point them out because we've already kind of snuck these in. So if we talk about things like dorsal, we're saying that something is towards the back of the body. And so if we look at the image on the screen, it shows the head. That's sort of the back of the head. I will point out that humans and these terms is actually a little bit problematic because most of the time, these terms are used on creatures that are on four legs. So if we were bent forward, that's what the terms are applying to. So for a lot of times when we talk about something being towards the back or towards the front, it's not the same way, the sort of up and down that we'd expect in a four-legged animal. So if you've learned these terms in relation to other species, just keep in mind that our orientation is sort of on that assumption that we could be on four limbs. So when we say dorsal, that's towards the back, so the back of our head, the back of our body. Ventral meaning towards the stomach, so on the front side. We can then talk about, I suppose we should talk about superior and anterior because they're close but not identical to dorsal and ventral. Superior meaning above and anterior meaning below. So with our orientation of our heads, superior and anterior gets awfully close to dorsal and ventral, but not quite. Which is why that figure right there is very handy. So you can see that there's a little bit of a tilt between dorsal and superior and ventral and anterior. So just keeping those in mind. Or, sorry, I'm meaning to say inferior. Oh dear, mornings. Superior and inferior, above and below. I apologize. And then we can have anterior and posterior, which are sort of towards the front and towards the back, which again are slightly off from dorsal and ventral. So this is a really handy little diagram to have around. If you are someone who is visual like I am, I would usually print this diagram out and keep it handy in case there's, say, a line of text that refers to a structure being superior or inferior to something. This is kind of a nice cheat sheet sort of slide. And then we can also talk about something being lateral or medial. Lateral meaning sort of towards one side or the other, medial meaning towards the center. So we can combine all of these terms to give us a concept of where things are. And this is here because a lot of the terms describing where different parts of the brain are will describe them relative to other parts of the brain. So if something is above another part of the brain, we might describe it as being superior to that part of the brain. And it doesn't mean that it's better than, it just means that it's above. Or it could be anterior to, meaning it's closer to this side than that side. And so this is what's useful there. We also have a second thing that's a little bit useful at the bottom for letter B that shows us different kinds of slices that you can view the brain using. Again, this is more of a for reference, but we have typically 3 different ways that we will cross-section a brain. This was originally used for terminology when you actually would cut open a brain or slice a brain to view it. Nowadays, we can get these kinds of slices using brain imaging techniques. So you can have an MRI that will give you something fairly similar. So it doesn't have to be a physical slice, but we still use the same terminology because the terminology gives us orientation information. So if we talk about a coronal slice, that's coming through this way on the brain. And so a lot of the images, exactly like the one we saw with our gyri and sulculi, that's a coronal image. So it's giving us that front-on or back-on view. We also have a sagittal, so if we're looking at it from the side, or we can have a horizontal, sort of looking at it from the top or bottom. So those are good planes to note. And that's another thing to keep in mind, that the brain is a three-dimensional structure, and depending on how we slice it, that will affect the shape of the diagrams that we're looking at. So for example, our next slide, we're going to be looking at a sagittal view of the brain, so sort of looking at it from the side. All right, so this is where we're getting into the different lobes. There are 4 lobes. The divisions are kind of arbitrary, and there's a little bit of communication in between things and stuff like that, but typically the lobes are named after the name of the skull bone that covers it. So that's how they got there. We just remember what they are from here. So we have the frontal lobe, that piece at the front, and it is located anterior to, or in front of, our central sulculus. So that's a sulculus, a line that goes this way, sort of from our ears across the top of our head. And that central sulculus then separates the frontal lobe in front of it from the parietal lobe, which is just behind it. We can also talk about the frontal lobe being superior to or above the lateral fissure, which sort of runs this way on the brain. And our frontal lobe, we'll talk more in depth again about its functions, but the bases are, it seems to be pretty complex, and that's where a lot of our interconnected stuff goes on. That's where a lot of the complex cognition that makes humans quote-unquote special would occur. A large part of our frontal lobe is concerned with movement and control of movement. So that's one of the things that we'll focus in on in just a minute. We will also talk about sort of our parietal lobe, that band there, the occipital lobe, the one at the very back of our skull, and then the temporal lobes, sort of at our temples, sort of each in order, but we're starting off with our frontal lobes. And so if we go to the full size of the image that zoomed in version came from, we can see a little bit more of a detailed diagram here, and they have different separations naming the different areas. So you've probably already heard about Broca's area and Wernicke's area, the visual cortex and the auditory cortex. We can talk about the motor cortex and the sensory cortex. So we have lots of different things that we can talk about. And again, we're going to go through each of them sort of one at a time. So starting with that motor cortex. So that is located in the precentral gyrus. So just ahead of that central gyrus. So where that central sulculus is, it's the lump right ahead of it, or the gyrus right before it. And so it's going to be involved in voluntary or non-reflexive movement. So that's movement that we are consciously making, not automatically occurring movement. When we talk about reflexive movement a little bit later on, that actually tends to happen more at the level of like the spinal cord, because it's a reflex arc and doesn't necessarily need the brain's involvement before it happens. We also have some stuff that we are not consciously aware of that's controlled in the brain stem, things like breathing. But what we care about here is stuff that we intend to do. So voluntary movement. And like we said, this is where we see that one side controls the opposite side of the body and vice versa. So the right side of your brain controls the left side of your body. Left side of your brain controls the right side of your body. And you'll usually see a representation of our central motor cortex mapped out onto a little like human figure that's all distorted to show you which parts control which parts of the body. They call this a homunculus or little man, and the proportions are all stretched out weird because different parts of our body have sort of larger or smaller parts of the motor cortex depending on how much dexterity they require. So you'll notice that our hands, which are very dexterous and involved in very complex tasks, take up a very large part of that cortex. So a lot of that is devoted to allowing us to move our fingers individually and move in very particular ways. Things like the knees or the hips have much smaller areas because they don't have to have as much dexterity or control. And then we have sort of most of the body, but then the face and all of that stuff is more on the sides here. So on the top is control of most of our body, and then the face and the mouth tend to be on the sides. And so this mapping onto this area, this isn't meant to be precise or exact. This is just general tendency of where these tend to be located. And conveniently, these also tend to line up with things that are usually associated together. So all of the fingers are in one place. So if you're trying to move 2 fingers at once, you're still activating pretty much the same area. And this motor cortex actually sits right next to the somatosensory cortex, which you might also see called a sensory cortex. So that's the one just behind the central sulcus. That's the gyrus just behind it. And that's where we get incoming information from all of these areas. So again, things that tend to be used together are located close together to reduce transmission time. So if we get a sensation about our hand that's relevant for our hand movements, it only has to travel from sort of one side to the other through those tracts to make sure that that information is communicated. So those are things that we want to keep in mind, how things are moving. And yeah, we've talked about more complex control. Perfect. Okay. The next thing that I wanted to talk about, because we're not going to move into that parietal lobe just yet, we're going to stick with our frontal lobe and talk about Broca's area. So Broca's area is the region that controls speech production. So this is where we can form words. So it contributes to grammatical structure, and it helps control the motors, or the motor control that we need for speech. So Broca's area is what is involved in helping us put together all of the movements and all of the particular sounds that we need to make to produce speech. And it's interesting, I'm actually doing a lecture in one of my intro classes all about language production. And that text gives a little bit more information about Broca's area. And so there was a man who had damage to this part of his brain. I believe he was kicked by a horse. And so Broca was the doctor or the physician who was attending him. And once this damage had happened, he was kind of okay at first, but then his performance got worse and worse and worse. And eventually he could only say one word. And that word was tan, which I thought was very strange. But that's what now the literature refers to this man. They call him Tan. And so they had to wait until Tan passed away from natural causes, and then they looked at his brain, and he had localized damage specifically to this part of the brain, to Broca's area. And so they made that assumption that because the damage was right in that area, and because it had such a severe impact on his ability to speak, that must be what was responsible for putting together all of the parts needed for speech. So it was kind of an interesting case study, and in all of the other times I've taught that sort of topic, I've never seen that story, so I thought I'd share it, even though it's not from this text. We have another specific case here of that hemispheric lateralization, where one side has sort of a different function than the other. And that is that language activity is typically controlled in the left hemisphere in most people. So about 9 out of 10 people have a left hemisphere centralized language control. And that doesn't mean that the remaining sort of 1 out of 10 is a right centralized. A lot of people, or I guess the other proportion of people, also tend to have sort of a diffused language control. And we typically have evidence for that in people who suffer strokes in Broca's area. And some people, even though the stroke is in the exact spot where it should impair their language function, some people are fine. And that usually tells us that their language control wasn't lateralized. It was actually shared between hemispheres.
This is kind of a very hypothetical question, but do you think possibly that girl who has a smooth brain, the reason why she functions so well is that the regular functions on the outside of the brain were maybe in other cases?
As a thought exercise, probably. I don't actually know if we get I'm sure we get a lot into it, but the brain is very plastic, which means that it can change function as required. So a lot of times we talk about like the standard location or standard function, but those areas can be completely reassigned. And if we ever get to sort of close up look at, say, people who are born blind, they don't need their visual cortex for visual anything. So that part of the brain can actually be co-opted to do other things. And so it might be that her brain prioritized tasks that were essential and kind of dropped tasks that weren't. But I don't know how they could go about studying that. It definitely makes me wonder, especially now that it's a lot easier to do like functional MRI or any of those ones where you can actively look at the brain and what it's doing in real time. Not too long ago, we were stuck with like still images or like hoping for the best. But yeah, a lot of the more technological studies nowadays give you real time, like what's the brain doing as it happens, which is really cool. So another one of those interesting, I don't know, but maybes. Okay, yeah, I think that's what I wanted to say about Broca's area. Usually we talk about Broca's area and Wernicke's area sort of back to back, but the way that our text has organized this is they want to talk about that frontal lobe first, and then we'll move to the other lobes. So you can remember that Broca's area and Wernicke's area tend to be sort of paired, at least conceptually in people's minds, because Broca's area is all about speech production, and Wernicke's area is all about speech comprehension. But we can revisit that when it becomes relevant. All right, so then we can talk about our prefrontal cortex. So that's the stuff that is towards the front. And again, this is basically a general term talking about the things that aren't our motor cortex or Broca's area. It's the things that are forward of that. And so this is a region that seems to be involved in planning and organization type behavior. That's going to activate when we're making decisions or changing our behavior based on potential consequences. If you remember the case study of Phineas Gage, he was the man who was struck in the head with a railroad spike. It damaged his prefrontal cortex, and he lost the ability, or his ability to evaluate consequences changed. In addition, his personality changed, his temperament changed, so all of that seems to be seated in that prefrontal cortex. And so that's one of those, it seems to be involved in a lot of things, and we'll talk again throughout the course about case studies where people have had damage to that area. Yep.
Sometimes when people want people to have to move, sometimes people talk about this life being happy as they search for more knowledge to have it. Do you think is the global area happiness of like
it might be. That's kind of an interesting idea. And this is one of those where I don't actually know off the top of my head, but we can kind of think logically about it. One of the cool things with language learning is depending on when you learn a language, it might get stored in different parts of your brain. So if you learn multiple languages when you're young, usually under the age of 5, but we seem to have kind of a flexible period up till about puberty. But if you learn that language young, the rules for that language, the grammar, the structures, the syntax, all of that tend to get stored in that like language part of the brain. However, if you pick up a language later in life, you store it somewhere else. And it kind of depends on where there's space for it. And that difference or that diffusion actually affects how you access those languages. So if it's like a language that you learned very young, it's a lot easier to switch between them because they're in the language center. So you don't have to go elsewhere in the brain to find it. It's not like you have to sort of route through different parts of the brain to translate, because you can think in each of those languages. But if you've learned it later in life, you would have to go from your language center, usually through your primary language, to wherever that second language is stored, and then kind of come back from there. And that's why for people who've learned languages later in life, it tends to take a little bit longer, and they might not have as smooth of a transition. But it's another one of those interesting things that's probably beyond the scope here. But yeah.
Oh. And can I give a follow-up question to that? So if you grow up bilingual and then you try to learn a third language as an adult, would it be harder to learn that third language?
It would depend on how it relates to the earlier languages. If they have the same root, it's not too bad. And I'm not an expert on this, but I believe that if they have similar roots, it makes it a lot easier because you can draw on your sort of existing understanding. So say you know French, but you're trying to learn Italian, because they have similar roots, you'd be okay. But if you tried to learn Icelandic, I don't know, then you'd struggle because it's a very different root. And I've actually, this is just sort of a N of one, I took French classes when I was younger. I took French all the way up to grade 12, but never did anything with it and dropped it for years. But with the pandemic, I started taking French again, and a lot of that stuff is still stored as instant recall because I learned it from 5 years old. So it's really weird, even though I was never fluent in it, having those rules and the structures preset in the language part of the brain makes it a lot easier than someone who maybe didn't have that same exposure, which is really, really cool. And it kind of makes me think of another sort of tangent, because we're right near the end of what I wanted to say anyways. But there's some discussion about cognition and that idea of maybe personality being tied to which parts of the brain are being activated. But the way that you talk about things, or the different language structures, are thought to also affect how you process information. So for example, the Russian language has more words for blue, so they have more words that differentiate between sort of light and dark blue, whereas English doesn't have that many. And so Russian speakers tend to perform better on color distinction or color matching tasks, especially where blue is related, versus English speakers. So there's some idea of how we think about concepts as related to the language that we speak, and that affects our perception of things as well. And so I'm wondering if our perception of stuff being shaped by our language might also affect sort of how we're thinking and then maybe our personality at that time. But yeah. So I think that Broca's area is also involved in writing, but I'm not positive. Because I think it's all about sort of taking the meaning and making it into the words that it needs to be. But it's a lot more complex than we really understand. Yeah. And you had a question as well?
Yeah, I totally forgot what it was.
Okay, no problem. Yeah. So it's one of those really interesting, complex things, and it's a very cool topic, but it gets well beyond our understanding quite yet, which is rather frustrating because I would love to know the answers. But I figured we'd end up off on a couple of tangents for this topic. So that was all I prepared for slides. What I'm going to do next time is we'll move on, talk about sort of the rest of our structures and progress through the other lobes. I'm assuming that we'll finish off our forebrain in the next lecture, and then we'll move on to sort of the rest. But the central nervous system is a big one, so we'll probably break it up as much as we can. But I will leave you guys there, and then we'll pick up on Thursday.